Hello, everyone. My name is Alexandros Marios Sofias, and I'm a postdoc at Professor Lammer's lab in Aachen University. And before I start with my talk, I would like to thank SemiLab for inviting me today for presenting our latest project. So what we did is that we used optical imaging at different levels of spatial resolution to identify the biodistribution profile of a polymeric micelle. And to give a short summary of what I will present today, we used a polymeric micelle labeled with fluorescence, and we injected that intravenously in mice bearing 41 tumors. And the first thing we did is to scan it at organism level with an optical imaging scanner, and then to continue with histological analysis and microscopy to identify the biodistribution of the nanoparticle at tissue and cell level. And the nanoparticle we used is developed by Crystal Therapeutics and is based on the CRIPEC technology. And at the moment is in clinical trials, phase 1 and phase 2. And if you are interested, you can find more information about this nanoparticle at the website of the company. And for whole body imaging, we used an organism level scanner developed by MI Labs, and is a combination of microcomputed tomography together with fluorescence tomography. And the innovation of this system is that we can have a 3D visualization of the entire mouse by using CT. And based on that, we can have organ segmentation. And then combine it with fluorescent detection, we can identify where the nanoparticles are located. Then by applying volumetric and absorption corrections for each individual organ, we can have a very significant advantage over traditional two-dimensional optical imaging systems that can allow us for quantification of fluorescence. And this is how the scanner looks, and this is in our lab. So we take the entire mouse and we put it in the scanner, and then we perform sequentially CT and then fluorescence scan. And the first experiment we did is to inject a small fluorophore, a small molecule, such that we identify how this small model drug will accumulate in the organism. And we can see that by scanning at multiple times over 2 days, that at early time points post-injection, the nanoparticle accumulates a lot in the liver. Then at later time points, we see that the nanoparticle has almost been cleared out of the organism entirely, and therefore is not capable of targeting the tumor anymore. And then the image changes completely when we inject our nanoparticle with the same fluorophore attached on it. And we see that early time post-injection, the nanoparticle accumulates in the liver in lower extent. But what is very important to observe is that at later time points, at 24 and 48 hours post-injection, we still see a lot of signal from the mouse, meaning that the nanoparticle has long circulating properties. And then we can very clearly see this time that the nanoparticle can achieve very high tumor targeting. As I said at the beginning of my talk, this technology gives us the opportunity to quantify fluorescence. And this is what we did. So you can see on the top line with gray, the quantification of the free fluorophore, and on the bottom line with red, the nanoparticle accumulation. And as we can appreciate, the tumor targeting is very strong when we use the nanoparticle. And if we focus on the heart, for example, we can see higher circulation half-lives. By summarizing these table in this nice heat map, we can see how nicely the profile changes overall. And we see that everything is shifted towards tumor targeting when we use the nanoparticle. And the nanoparticle, because it circulates longer, it can even achieve better targeting in distant tissues that are hard to be targeted, for example, spinal marrow. And the outcome is that by targeting the tumor and using this nanoparticle, we achieve almost a 20% of injected dose per gram of tumor at around 24 and 48 hours post-injection. Then we continue by ex vivo imaging. So after 48 hours, we take the mouse and we sacrifice it and we collect the organs. And we are preparing them for ex vivo imaging. The reason we do that is that we want to corroborate our in vivo imaging data with ex vivo imaging. And you see again, we are preparing everything and we are ready to scan. We insert everything on a 2D fluorescent reflectance imaging scanner, and we generate the following images. You can see on the left side how the free dye looks like. We see a lot of accumulation in the kidneys. This is how small molecules behave typically. And then on the right side, we see how the nanoparticle behaves. So it targets the liver, but above everything, it targets the tumor. Very high quality of targeting that confirms what we observed in vivo. And then, of course, we can quantify the data from ex vivo imaging, and you can see in these three different panels that we confirm with numbers that nanoparticles are able to target the tumor. And a nice, interesting information here by comparing the first and the second figure that shows the average fluorescence in an organ and the max fluorescence in organ. And if we focus in liver, spleen, and tumor, we see that even though the axes, they do not have the exact same unit, the pattern is similar between the mean and the max. And this is indicative for homogeneous distribution of the nanoparticle in the tissue. And speaking about distribution, we come to our next step, which is to investigate the accumulation of nanoparticle on tissue level by performing histology. And at the beginning, we perform histology on tissue level, so more macroscopic. And on the top line, you can see slices from tumor, liver, and spleen. And we see the accumulation of nanoparticles, which we see with yellow, and with light blue, we can see the nucleus of the cells. And on the top line, it's all nanoparticles, and in the bottom line is only the endocytosed nanoparticles. And by quantifying these images, we can understand how many nanoparticles are localized inside or outside cells. And we observe the same pattern. So approximately 67% of nanoparticles remain extracellularly, and this is preserved among the different tissues. Our next step is to focus deeper on our histology, and then by using specific markers for the different cells located in the tissue microenvironment. We can see now here an example of tumor that we stain on the top line, for example, for CD45 positive leukocytes, CD31 positive endothelial cells, and we still have the nucleus appearing as blue and the nanoparticle signal appearing as yellow. On the bottom line, we can see more or less same type of analysis, but instead of CD45 marker, we used a macrophage marker, and then we take these images and we can get some good numbers out of them. And on the left side, you can see the composition of the tissue in the different cells. We see, for example, that in tumor, approximately 70% of the tumor microenvironment is composed of cancer cells, approximately 24% is comprised by immune cells. Macrophages are the most dominant population as expected. And on the right side, we can associate the amount of the number of nanoparticles that is localized inside of these different type of immune cells and cancer cells. And by doing the same approach, by following the same approach in tumor, liver, and spleen, we saw that approximately 33 to 50% of the intracellular nanoparticles are localized in F480 positive macrophages. And then to go again to my summary slide, we have, by analyzing our data, we saw that by using these nanoparticles, we are able to target tumors in on a very efficient way. We see a 20% of the injected dose per gram of tumor was a very good result for us. And also we saw very low off-target toxicity, since the nanoparticle has the preference only to localize mostly in the tumor. And then by performing microscopic analysis and histology, we saw that at tissue level, the nanoparticle, the majority, 67%, remains extracellularly. And this is promising because we can see that by the prolonged release of the drug, the free drug inside the tumor microenvironment can be taken up by neighboring cancer cells. Then, by focusing on the individual cells that are engaging with the nanoparticle, we see that the majority of the intracellular nanoparticle is inside macrophages, and this is a nice possibility for us in order to develop immunomodulatory strategies. And this summarizes my talk. Our publication on the Journal of Control Release is available. You can feel free to look at it for more details if you are interested. And then if you are interested in our work, you can visit our website at the Department of Nanomedicine and Theranostics in the Institution of Experimental Molecular Imaging in Aachen. Thank you very much.